Okay, I will go quickly on the introduction because everything has been more or less really well introduced by the previous speakers. Just one very important thing, just to remind again, the inactivated or latent provirus is a stalled promoter. So there is a stalled promoter, and to activate that promoter, one needs P-TEFb, and we are interested in studying P-TEFb, what activates P-TEFb and what represses P-TEFb. We've heard about how Tat works. So P-TEFb in the cell is regulated, so it appears in two flavors. In an inactive complex where its kinase activity, the CDK9 kinase activity, is inhibited by the hexene protein, and this complex is in an equilibrium with a free or active state where P-TEFb is recruited by largely by BRD4 to activate promoters, to promote pause release of stalled promoters. Tat has been shown in vitro to displace hexene 1 from cyclin T1 and thus generating free P-TEFb, which will then be recruited by Tat to activate the viral promoter and produce the virus. The inactive 7-SK-SNRP, so the inactive P-TEFb complex has so far been fairly well characterized, but very little is known about the active P-TEFb or elongating P-TEFb complex. And our aim was to characterize, to purify, and to study this active P-TEFb complex. And our strategy was to co-purify that complex with Tat, because what we know about Tat is that it forms stoichiometric complexes with P-TEFb, and Tat recruits P-TEFb to an already engaged RNA polymerase molecule, and thus one should be able to co-purify active P-TEFb by purifying Tat. Our strategy is fairly old school. We create a stable cell line expressing Tat in fusion with two peptide-elutable tags, and purify Tat by double affinity chromatography from HeLa dignum nuclear extracts. So this is the result of the purification. As has been previously shown, you have stoichiometric amounts of Tat and P-TEFb subunit CDK9 and cyclin T1, as well as different classes of elongation factors. So we know about P-TEFb, but ELL and PAF1 promote elongation by different mechanisms. And we also found some of the most common MLL fusion proteins within this complex, which have been linked to transcription, but the role in transcription is not very clear. Somewhat surprisingly, we found stoichiometric amounts of LARP7 and MEPC, which are 7-SK-SNRP subunits within the Tat-purified material, as well as 7-SK RNA. So next, we asked the question whether any of these associations fall into one or several complexes. For this, we subjected single purified Tat, flag purified Tat, to glycerol gradient sedimentation, as well as reciprocal IP against subunits we identified previously in the previous slides. And I will only show you the results of the glycerol gradient. Basically, simply put, you will find two peaks. First peak here containing the PAF1 complex subunits, the ELLs, as well as the MLL fusion protein partners, and a second peak containing the 7-SK-SNRP subunits LARP7 and MEPCE, as well as the 7-SK RNA, suggesting that there are two complexes. Highest activity was associated with the first complex, and for the remainder of the talk, I will focus on this first complex here, which we call TATCOM1, and which we believe is the active or elongating form of P-TEFb bound to Tat. So these associations occur in a Dürker T cell line, as well as so far tested for the ones we have tested so far in PBMCs. So first of all, active P-TEFb and 7-SK-SNRP subunits are induced upon T cell activation. So you can look at CDK9 here, for instance. It's less induced than, let's say, hexene 1 or cyclin T, or these new factors, AFF4. The same is true for AF9 and AFF4, as well as ELL here. ERK as a control, and both active and 7-SK-SNRP bound P-TEFb complexes increase with T cell activation. And the last experiment we did on PBMCs was a GST pull down with Tat on extracts derived from PBMCs, suggesting that TATCOM1 and TATCOM2 both form in those cells. This is fairly preliminary, this slide. Okay, so the formation of these complexes is P-TEFb dependent. I cut this short. We have a lot of proof for that. I show you this experiment here, which shows that if you take away CDK9 from TAT complexes, you almost abolish CTD kinase activity of that complex. That is important because we next ask the question, why does TAT form such a complex? And the most easy way to address this question is to compare kinase activity. So we take the same gradient as I showed you before. You have this, in this very first fraction, you have only P-TEFb, so cyclin T1 and CDK9 alone bound to TAT, or the fraction 7 where you have the entire complex, and we compared, we leveled, we normalized for CDK9 levels and measured CTD kinase activity, and there is highest activity associated with TATCOM1 as compared to core P-TEFb, which suggests once again that this is really the active form of that complex. So more precisely, one of the subunits, AF9, we could show that it is required for optimal CTD kinase activity as well as for ELL binding to the complex. A very important message from this work is that this complex exists in the cells in the absence of TAT, but what TAT is doing is it increases the association of these different cofactors with P-TEFb. So on the left here, it is difficult without a laser pointer, but so believe me, just so if you IP cyclin T, also in the PBMCs, as has been shown previously, these were endogenous IPs, you co-IP all these factors, but in the presence of TAT, you IP more, for the same amount of CDK9, you IP more of these positive acting factors on the TATCOM1. So somehow, we don't know how, but somehow TAT increases the concentration of this complex in the cell. So then lastly, these cofactors are required for TAT-mediated transactivation, and more precisely, the AF9 subunits, which we showed is required for optimal CTD kinase activity, is required for TAT-induced elongation by measuring the ratio between distal and proximal transcripts. Importantly, TATCOM1 subunits are present not only at the promoter region, but are present throughout the gene, which strongly suggests that TATCOM1 participates in transcription elongation per se. So a summary slide. So TAT forms at least 2 biochemically and functionally distinct complexes. TAT induces the formation of a complex composed of P-TEFb and this leukemia module, as well as PAF1. I forgot to mention, we talk about a module here because TAT increases the association of these proteins with P-TEFb, but not the associations between them. TATCOM1 is involved in transcription elongation from the HIV-1 promoter, and AF9 is required for optimal CDK9 CTD kinase activity, as well as ELL recruitment to TATCOM1. One thing we are very interested in is finding mechanisms and signals that regulate this equilibrium between core free P-TEFb and this active or elongating P-TEFb. Which signals pathways are required to form this complex, or which are involved in the expression or stability of these different cofactors. And one way to explore this is, of course, to try to understand how TAT does so. So lastly, I would like to thank my PI, Monsef Benkirane, and Reservoirs for tutorship and lots of help. Nadine Laguette and Ahmad Yatim, who helped very much during this work, as well as Chang Zhu from the University of Berkeley and the funding agencies. Thank you very much.
Thank you very much. So we have time for a couple of questions. So while people are thinking about what to ask, I will ask a question of clarification. What is AF9? Because I don't think you defined it. What is AF9? Could you tell us?
Oh yeah. So okay. So AF9 is part of the active. So TAT forms two complexes. One contains 7SK SNR&P subunits, and the other one contains factors which we believe have a positive role on HIV transcription, and AF9 is one of these proteins. So it's part of TATCOM1. It's a TATCOM1 subunit.
Okay, so that explains the AF9 knockout. And you said that there are two complexes, in one TATCOM1 and one TATCOM2, and you focused on TATCOM1. Why do you think TATCOM2 is not equally important?
I don't say that. It's just the way it happened.
Okay. Other questions for this presentation?
I have a question. I don't know if it worked. Well, what about the TATCOM2 complex in the presence of TAR?
Sorry?
Is still the 7SK associated with the TATCOM2 complex in the presence of TAR?
So I'm not sure I understand. So what happens in vitro if I add TAR? I don't know. I don't know. We haven't done that experiment.
So what's next for you in terms of the siRNA experiment against TATCOM1? Is there a follow-up to this to perhaps develop some extra special strategy based on this nice initial result?
Yeah, so, well, what we mostly focus on is how that complex is formed. So that complex, so the various subunits and P-TEFb, they do associate it in cells in the absence of TAT, but TAT increases the association of these cofactors, which are required for optimal P-TEFb function. And what we focus now on is mostly how this complex is formed, which cellular signals are required for this.
I was wondering also, have you looked at the possible differences between the two different forms of TAT, 72, TAT 101, and binding to the two complexes?
Oh, 72 and 101?
Yes.
No, I haven't done that.
And have you searched for mutants that might kind of differentially allow TAT to cement this interaction? You have mutants that, TAT mutants that actually disable its ability to increase the interaction in TAT1.
Yeah, so we've done that. So in the proof to show that P-TEFb is required for formation of these complexes, we use the C22G mutant. So a C22G mutant cannot form these complexes, for instance.
C22G is also involved in cyclin T binding. I was thinking more of specific mutants that have been known around, with really not well-defined mechanisms like K41, for example, or other mutations.
Anyway, we have to move on.
I'm not sure. For sure, the C22G cannot form any of this. That's in the paper.